video, we are going to be looking at the final pathway of the complement system, the alternative pathway. If you've not seen the classical pathway and the lectin pathway, do well to see them because I've covered it in previous video. Now, the alternative pathway of the complement system is an antibody-independent pathway, meaning it does not require an antibody to get activated, right? This pathway gets activated by the attachment of C3b to a microbial surface, so it doesn't require an antibody. What happens in the alternative pathway? Let's look at it. Remember that we've said that the complement system is made up of a group of proteins C1 to C9. C3, one of the complement proteins, is found in the blood, and this C3 is continuously undergoing spontaneous hydrolysis, and this hydrolysis produces C3b. Now, this is where it begins. This C3b attaches to a microbial surface, C3b. It attaches to a microbial surface. If there is no regulatory mechanism to inactivate it, it activates the alternative pathway. What happens next is that factor B comes and attaches to this C3b. It's a complement protein. It comes and attaches to this factor B. It comes and attaches to this C3b. So it becomes C3b. To make this attachment to the microbial surface stronger, it happens in the presence of magnesium, which is a cofactor. So this covalent attachment of factor B to C3b on the microbial surface happens in the presence of a cofactor, magnesium, to make the attachment stronger. After factor B has attached to C3b, the next thing that comes in is factor D, which is an enzyme. Factor D, an enzyme that cleaves this B into BA and BB. Do you understand? It cleaves B into BA and BB. So remember, in the complement system, the A is the smaller fragment, so this diffuses away. B, this now becomes, after the cleaving of factor D, it becomes C3BBB. Are you seeing it? C3BB. This is also known as the C3 convertase. It is the protein that will cleave C3 into C3a and C3b. Do you understand how it's going? Now, let's continue. After the cleaving of C3a to C3a and C3b, this thing now becomes C3b. It becomes C3b, BB, and C3b. This is known as the C5 convertase. C5 convertase cleaves C5. It cleaves C5 into C5a and C5b. Like we've said in previous video, C5a is a powerful inflammatory molecule. It also acts as a powerful chemoattractant. So it goes ahead to attract other immune cells to the site, while C5b initiates the formation of the membrane attack complex. So the C5b binds to this, becomes C3b, BB, C3b, C5b. C6 is activated, it joins it. So at the end of the alternative pathway, what you'll be having is C3b, BB, C3b, C5b, C6b, C7b, C8b, C9b. This is known as the membrane attack complex of the complement system. So you see that both for the alternative pathway, the lectin pathway, the classical pathway, from the cleaving of C3, the reaction becomes the same. It leads to one final outcome, which is the activation of the membrane attack complex. And we've said it in previous video that the function of this membrane attack complex is to puncture holes in the cell membrane of the microbe and lead to cell lysis. So there are 3 points you should note in the alternative pathway. It starts by So there are 3 points you should note in the alternative pathway. It starts through the hydrolysis of C3 to yield C3b. C3b binds to the microbial surface. Factor B comes and binds to it in the presence of a cofactor, which is magnesium, to help it to covalently bind to the surface. The next thing that happens is factor D, an enzyme. It comes and cleaves that factor B into BA and BB. BA diffuses away. BB remains bind to the C3b. So it becomes C3b-BB, which is the C3 convertase that cleaves C3 into C3a and C3b. C3b goes ahead to be an inflammatory molecule. C3b binds to the reaction. It now becomes C3b-BB, C3b, which is also known as the C5 convertase. So that C5 convertase cleaves C5 into C5a and C5b. So the reaction continues like that. You should also know that the alternative pathway is antibody independent, right? The mannose-lectin pathway and the classical pathway have similarity because they both involve the cleaving of C4 and C4 While in the alternative pathway, the reaction starts with C3b. So note that in the alternative pathway, the reaction starts with C3b, unlike in the classical pathway and in the mannose-lectin binding pathway. If you have any question, drop it in the comment section. I'll be answering your questions.